Thank you for joining us. This is a recording of Professor Christopher McQuaid's excellent lecture on marine biology, and I'm sure you'll find it very useful. Before I get going with the recording, you might like to have a look at some of the forthcoming events that the Centre for Postgraduate Studies will be offering, and we look forward to seeing you at some of those too.
And how we do it. So the first question for me is why study marine biology? And my answer actually is curiosity. And that's very much a personal answer, because I do go along with something which the past president of this country, Thabo Mbeki, once said, which is that this should not be a country which only aspires to clothe and feed itself. I think that all the major civilizations of the past have been characterized by strength in some aspect of science. So it might have been the ancient Greek formulation of the natural world, their philosophy. It might have been Mayan astronomy. It might have been Arab mathematics, or it could have been Roman engineering. But all the major civilizations were characterized by a love of knowledge, the acquisition of knowledge, and strength in some aspect of science. And that's what motivates me. But of course, there's the argument that all the research is funded by taxpayers' money, and so there should be some more direct benefits to society. I can address that by saying, yes, well, we have curiosity-driven science, which to me is important because there are government institutions which deal with applied problems. We have, for example, in marine biology, we have the Department of Water Affairs, we have we have marine and coastal management, and there are huge organizations which are designed to deal with practical problems. And the only organizations which are mandated with the task of doing pure research, or so-called pure research, blue skies research, curiosity-driven research, those are the universities. So I feel that the universities really should emphasize curiosity-driven research. But as I said, there is an argument to be made that there should be some direct benefits to society. And so it's very easy to make an argument that if you study marine biology, it's an important contribution to the management of natural resources. It's very important for conserving biodiversity, which of course underpins human society. And it's also important to understand how present-day ecosystems are going to cope with climate change. So those are the sorts of reasons for why I study marine biology. And then we could ask, well, what's special about marine biology in South Africa? And the answer is that actually, although most people don't appreciate it, this country is one where three completely different oceans meet. We have the Atlantic Ocean to the west of the country, we have the Indian Ocean to the east of the country, and we have the Southern Ocean to the south of the country. And we conduct at Rhodes University, we do research in all three of these oceans. I'm going to start by looking at the Southern Ocean. And if you're not familiar with the Southern Ocean, this is a map of the world viewed from the South Pole. So that's the Antarctic continent. You can see Africa to the top right. We can see South America to the top left, and Australia to the bottom right. And all the ocean between those continents and the Antarctic is what's called the Southern Ocean. And to give you a sense of scale, this ocean is roughly the same size as the continent of Africa. So it's a very, very big place. We do a lot of research which entails going right down to the Antarctic continent, and to sail from Cape Town to the Antarctic continent will take about 10 days or so. So this is a very large place. It's also a place which is particularly vulnerable to climate change. And that's important because, as well as being vulnerable to climate change, the Southern Ocean is a major driver of global climate. So there's an interaction between the change in climate influencing the Antarctic and the Southern Ocean, and the Southern Ocean in return feeding back into global climate. So understanding how the Southern Ocean will respond to climate change is really critical to understanding what our future is going to look like. And in the Southern Ocean, we do research partially, we do research around Marion Island, which is, as some of you will know, is a base about halfway between Antarctica and Cape Town, and it's a permanent meteorological base manned by South Africans. And on this island, there are enormous colonies of land-based animals, animals which come to land to breed. For example, we have penguins, we have elephant seals, we have albatrosses. There are all sorts of animals, and there are really enormous numbers of these beasts. They have to come to land in order to breed, and in the case of the birds, they have to molt as well and develop new feathers. And both molting and reproduction are energetically very expensive, and we've done a lot of work on trying to understand where the energy comes from to maintain these large populations of animals. And when I say large, there are literally millions of penguins on Marion Island, and these are some of the largest penguin rookeries on the planet. So there are enormous numbers of energy-consuming animals on these islands, and we ask where does the energy come from to support them? Some of them are, as I said, these penguins. Some of them are these rather magnificent albatrosses, and if you're not familiar with albatrosses, they are very large birds indeed, and they too are breeding on Marion Island. In order to study them, we do ship-based research where we go into the actual water column, and we do a lot of research based on this animal on the right-hand side of the screen. It's called krill. It's basically a shrimp-like animal, and it's a completely central feature of Southern Ocean food webs. Just about everything in the Southern Ocean depends directly or indirectly on krill. So it might be the great whales, it might be seals, it might be seabirds. One way or another, they all depend on these animals' krill. And they, of course, are very vulnerable to changing climate, loss of sea ice in the Southern Ocean, and rising temperatures. The sort of work that we do involves making measurements of the water column. For example, we measure chlorophyll, which allows us to estimate rates of photosynthesis by the plants in the water column. And we also use these things which are called bongo nets to catch animals which live in the water column itself. And we use dredges to bring up animals that live on the floor of the sea. And some of these are pretty strange looking animals, as Sue mentioned. In the top left there, we have a type of worm. It's called a polychaete. Here we have a squid. Here we have a rather large looking octopus. And here we have a very bizarre fish. I want to look at that a little bit more carefully because it's very typical of deep water animals. Deep water, in very deep waters, there's very little food. So the rationale there is if you encounter something and it's edible, you have to eat it. And it doesn't matter how big it is or how small it is, you have to be able to eat it. So some of these animals are extraordinary in that they are able to consume prey which are bigger than they are. So just pause for a moment and imagining swallowing something which is bigger than you are. And they can do that because they have a very extensible stomach. They have jaws which hinge to the perfectly wide open, and they have these rather snake-like teeth, which make sure that once they've got hold of something, it's not going to get away again. So there's some really very bizarre animals that live in the deep sea. The trouble with the Southern Ocean is that it's not for the squeamish. It's very, very rough. It's perpetually stormy. And if you're prone to seasickness, you don't want to be in the Southern Ocean. As well as working in the Southern Ocean, we do a lot of work around the coastline of the country. And if we look at a map of South Africa, you can see that the coastline is dominated by different currents. On the west coast, we have what's called the Benguela current. It's a very cold water current. It's full of nutrients, and that promotes a lot of primary production by phytoplankton in the water column. On the east coast, we have the Agulhas current, which is a much warmer current. It flows down around the coast and then turns back on itself. It's very saline, very low nutrient content, much lower productivity. And when I say that the Benguela current is cold, if any of you have ever gone swimming at Camps Bay, you'll know what I mean. It's really, really cold, maybe 9 degrees or so. If you go to East London, something like that, the temperatures will be more of the order of 20 degrees. And so these major differences in temperature and the links to different oceans, the Atlantic to the west and the Indian Ocean to the east, tend to result in the development of what are called biogeographic provinces. We have very different communities of plants and animals that live on different parts of the country. We have in the east, the subtropical Natal province running up into Mozambique. We have on the west, we have the cool temperate Namakwa province running up towards Namibia and into the south of Angola. And along the south coast here, we have what's called the warm temperate Agulhas province. And those different regions are characterized by different communities of plants and animals, which are as different as, let's say, a Karoo biome, a semi-desert biome, and the fynbos communities in the Western Cape. So these are very, very different groups of plants and animals. And if we look around the shore, we have very different different types of shores. As you can see, there's a huge variety of different types of shores. If we go to somewhere like the Transkei coast on the south coast of this country, we have these very flat wave-cut platforms on these hills that drop steeply into the sea and pocket beaches between these hills. Around the region of Port Elizabeth, we have extensive stretches of sandy beach. And if you go to somewhere like Cape Town, in the Western Cape, we have these huge granite boulders. So really very different environments in different places around the coast. So we ask what sorts of animals live in these different places. And as I said, they can be very different. On the West Coast and the South Coast, we have these very dense populations of rather large limpets. On the South Coast, we have reefs which are formed by sand reefs built by worms. So each of these little holes is the home of a polychaete worm. And on the East Coast, we have large numbers of zoanthids, which are a little bit like sea anemones. So really very different looking organisms in different parts of the coast. So we ask questions like, why are different communities so different on the coast? And will these different communities respond differently to stressors? So I want to ask, what sorts of things stress marine ecosystems? What sort of things threaten them? And I'm going to look basically at some anthropogenically driven stressors. And the first one I want to look at is invasive species. An invasive species is a species which has been brought into the country from someplace else on the planet. It then spreads, it competes with the native species, and basically can drive them to extinction. And I'm going to use an example of mussels. I'll use mussels as good examples of a lot of things. And here's a photograph of a mussel bed on the coast. And if you look carefully, you'll see there are all sorts of other things there. There's a little bit of seaweed there. There's a limpet over there. There's a very different type of limpet there. There's a third species of limpet here. And basically, if you dig deep into one of these mussel beds, you find that they support a really wide range of biodiversity. So mussels are important because they create habitat for other species and maintain biodiversity. And biodiversity is important because it helps the ecosystem to deliver the sorts of ecosystem services on which we as a society depend. Here's a photograph of a mussel, and it tells you a couple of things. First is that we spend a lot of time training mussels to settle in the shape of a map of Africa. And in case you're not quite orientated, we are here, or I am here, down in Grahamstown on the south coast. And it's a bit hard to see, but this is a mixture of these brown mussels, which are the local mussels, indigenous mussels called perna. I'll use the Latin name and the descriptive name. And these invasive mussels, which are blue, black mussels and called Mediterranean mussels because they come from the Mediterranean. And if we look around the coast, we find that on the west coast of the country, the invasive Mediterranean mussel Mytilus dominates the coast entirely. It has eliminated and driven to extinction a couple of species, native species of South African mussel. On the east coast, the mussel beds are still dominated by the brown mussel, Perna perna, which is an indigenous mussel. And on the south coast, we find that the two are in competition with each other for space. These animals need somewhere to live, to attach to on the shore, and space is often limiting, and they will often compete both with their own species and with other species in order to get space to live on. And on the south coast, we find that the invasive Mediterranean mussel is high on the shore. The indigenous brown mussel is low on the shore. And in between, there's a zone where they are mixed together and where they compete with one another. So if you go to one of these shores on the south coast of South Africa, you'll find blue mussels at the top of the shore, brown at the bottom, and a mixture in the middle. And we've done a lot of work on the interaction between the invasive and the indigenous mussels. So when you look at a mussel bed, there you are, there's a nice mussel bed full of brown mussels. But when you look more carefully, you realize that, oh, there are some blue Mediterranean mussels, and oh, there are some more, and some more, and some more, and so on. So all these invasive species are not necessarily conspicuous. These are doing the same thing in the sea that in South Africa, the acacias, the fir trees, the oak trees have done. They've been brought in from somewhere else and they've outcompeted the local species. Another important stressor for marine systems is overexploitation. And this to me is an interesting one. Here is a photograph from Mozambique. These are oyster shells. And you can see that there's enormous numbers of oyster shells being eaten by people in Mozambique. In South Africa, most of the exploitation of shellfish is targeted around mussels. And this has been going on for a very long time. Here is what's called a midden. And a midden is basically a rubbish heap for people who've gone to the coast, pick up some shellfish, eat what's inside, toss the shell aside, and the shells accumulate into these great big piles of middens. And this is an ancient Strandloper midden. It's probably thousands of years old. But this sort of exploitation of mussels is still going on. And the brown indigenous mussel I mentioned is particularly interesting in this context because it is amongst the very first seafoods eaten by mankind. So for millions of years, hominids had diets which were based entirely on terrestrial food, on terrestrial plants, terrestrial animals. And there's a site near Mossel Bay on the south coast of South Africa where one of the oldest middens that we are aware of contains the very first indications of the inclusion of seafood into hominid diets. And this is important because some of the minerals in seafood are believed to have been important in the development of the brain. And in this case, in this particular midden near Mossel Bay, the dominant organism in the middens, the dominant marine organism is the brown mussel perna perna. So this animal has been consumed for tens of thousands of years and is being eaten to this day in Southern Africa. In fact, it's the inclusion of seafood into the diet of mankind, which we think aided the ability of man to move out of our cradle in Africa, where we originated, and to spread around the globe. And you can see many of these routes of dispersion follow the coastline along here down the coast of the Americas. And this was basically made possible because if you were traveling long distances over protracted periods, you didn't have to hunt to find food. You didn't have to plant crops and harvest them. You could simply go to the coast and pick up some food. And so we believe that the inclusion of seafood also was very important in the ability of mankind to spread out of Africa to the rest of the world. If we think about exploitation of seafoods in South Africa, and we look at where human populations are densest along the coastline, we have, of course, the urban centers, Cape Town, Port Elizabeth, and so on. But we also have rather a high density population in what's called the Transkei region of South Africa. And if we look at levels of poverty, this is an area where not only are human populations very dense, but also there are very high levels of poverty. And high levels of poverty usually result in high levels of exploitation of natural resources. So here we see a lot of ladies down on the beach, a lot of women down on the beach collecting seafood. Seafood is free. Going to the shop and buying food is not free. So if you are economically struggling, it's the best thing you can do is to go to the beach and find some food there. Apart from the sort of economic and food aspect, this has a very important cultural aspect as well. You can see all these people here are women. Some of them are 9 years old, some of them are 90 years old, but they're all female. And this is a very important social interaction for these people, as well as being a way of finding food. But as you can imagine, this many people going down to the beach will exert huge exploitation pressure. And it can change ashore from looking something like this little inset, which is covered in mussels, to something like this main photograph where the shore is covered in seaweed. And this is a particular site called Tjelanyama on the Transkei coast. And I'm going to come back to Tjelanyama a little bit later. But the point here is that overexploitation has eliminated the mussels and driven the community from one dominated by animals which you can eat, i.e. mussels, to seaweeds which you cannot eat. And we do have long-term data which indicate that if you shift the community from mussels to seaweeds, it will stay and leave it alone. It will stay as seaweeds for at least 17 or 18 years. We don't know how much longer, but at least for that sort of period. So in other words, in the vicinity of 20 years. Once you destroy the system, once you've unbalanced the system, it doesn't just bounce back again. Stopping exploitation is not enough. So we can do two things to try and cope with this problem. The first is to mitigate the extent of exploitation. And here are two photographs, one from Morocco in North Africa, one from the Transkei in South Africa. And these are two ladies who are both collecting mussels, but there are important differences in how they're collecting them. First of all, you can see that the lady in Morocco is collecting enormous numbers of mussels. She's just filling up a sack. Whereas the lady in the Transkei is collecting a much more limited number of mussels. And this is because we have legislation in place now as a result of research setting a limit to how many mussels people are allowed to collect. The second thing is that if you look at the implements they're using, the lady in Transkei has got a really narrow bladed instrument. It's actually a screwdriver, which allows her to pick this mussel, not that one, that mussel, not this one, and select the animal she's going to take. The lady in Morocco has a much cruder implement, and she just scrapes up everything she can find. And the consequences of these differences are actually pretty conspicuous. This lady in Transkei is standing on a mussel bed. This lady in Morocco is standing on a shore which is dominated by seaweeds. And to get these mussels, she had to go very deep into the water. Eventually, she will drive the prey, the mussels, to extinction to the point where she can no longer collect any. So the first way we can mitigate damage to our overexploitation is to moderate the form of exploitation, how many animals are collected and how they are collected. And key people in this process are these two ladies with the suns over their heads. And these are monitors appointed by the local community to ensure that everybody is abiding by the regulations. So you can see all these ladies are carrying small containers because there's a limit to how many mussels they can collect. And at least this one on the extreme left, you can see she's got a very narrow blade that she's using to collect animals, to collect mussels. So we can regulate the extent of exploitation pressure, assuming there is buy-in from the local community. And without buy-in from the local community, this simply does not work. But despite this, we already have sites which have already been overexploited. And so the question is, as I said, if we leave them alone, they don't bounce back for the next something like 20 years. But what we can do is we can speed up the recovery of the shore by rehabilitation, active rehabilitation. And we do this using a very, very simple approach. What you see here is a photograph of a piece of irrigation piping. It's very coarse mesh plastic piping. It's been cut into a short length, about 40 centimeters long, and then it's been cut lengthwise. So that's half of a plastic pipe. We screw it down onto the shore on top of what's called bycatch. When people collect mussels and say, oh, this one's too small to eat, I'll throw it away. The answer is no. You put it under one of these with all the other bycatch. You put it underneath one of these pipes. You screw them onto the shore and you leave them there for a couple of months. After a couple of months, you take up the pipe, and what you have is a very crude little rough bed of mussels. Now, that's not going to do very much on its own, but that's just the start of rehabilitation. You can then put down lots and lots of these pipes. And here you can see a shore where there are lines of little mussel patches where the pipes have been taken away. Here's a pipe which is still in position. Here are some more that are still in position. These ones have been taken away, and you can see these little collective little patches of mussels. These will eventually grow and coalesce and expand and eventually result in successful rehabilitation of the shore. And here's Chelanyama before rehabilitation, and here it is again afterwards. So we can take a shore back from being covered in seaweeds, which you can't eat, to being covered in mussels, which we can eat. But the real issue is that rehabilitation works, but that's not the source of the problem. The biology is actually relatively simple. And the real problems are socioeconomic ones. 100 years ago, 150 years ago, people in many poor countries in Europe, like Ireland and Portugal, used to go down to the seashore, just like they do in Transkei today, and collect things to eat. And during the 2008 economic crash, in global crash in economics, we saw suddenly an increase in the number of people going down to the shore to collect seafood in Portugal. So really the real drivers of this overexploitation are socio-economic, or to put it another way, if we want to take care of our environment, we actually have to take care of our people. That's the real solution. Rehabilitation is not. Looking after people is the real solution. But that sort of borders on sociology almost. I'm, again, more interested in the actual biology. And in order to manage these things properly, you have to understand the biology. So we as a species, Homo sapiens, we are mammals. Mammals have internal fertilization. Mussels don't have internal fertilization. Mussels shed their sperm and their eggs into the water column. The male releases sperm into the water, the female releases eggs, and fertilization happens outside to develop a very, very simplistic, if you like, juvenile mussel called a larva. This is a tiny little organism. It's only about a fraction of a millimeter long. In fact, these are quite old larvae. When they first start off, they're about a quarter this size. And these things live in the water, and they have to come back to the shore to join the adult population. And you can imagine, if you're that size, you're not going to get from one place to another or get to the shore to settle by virtue of being able to swim. So it's really difficult to understand. Once these eggs and sperm are released into the water column, the egg is fertilized, it forms a larva. Where is that larva going to end up? And my analogy is quite simple. If you have a field with a known number of sheep in it, you know roughly how many lambs you're going to have next year. But if you have a mussel bed with a known number of mussels on it, you have no idea how many mussels are going to appear, how many juvenile mussels will appear next year. So the ways and the distances over which larvae disperse are really important to understanding the control of mussel populations. So let's say you want to design a marine protected area. And the brown bit at the top, this is meant to be a bird's eye view. The brown bit at the top is the land. The blue bit at the bottom is the sea. Here we have this nice green bit, which is protected. Nobody's allowed to collect mussels there. The concept is that these mussels will flourish, they will grow, they will reproduce, they will produce larvae. Those larvae will spread out into the adjacent areas where exploitation is happening and reseed the exploited shores. But how do you design your marine protected area? Let's say you decide to set aside 30 kilometers of coastline. Should it be a single block like this? Or should it be 3 10-kilometer blocks? Which is going to be more efficient? And the answer is, of course, lies in how far do the larvae disperse. And so that's the type of research which we've done a lot of work on. It's very, very difficult, but it's actually fundamental to understanding how to manage these natural resources. The next anthropogenic driver of change or threat to marine ecosystems is, of course, climate change. And we're pretty familiar with this. Temperatures are going to rise. Some organisms will be able to tolerate it. Some organisms will not be able to tolerate it. How will ecosystems change in the future? And I'm going to take a slightly different view on that to show how interactions amongst species makes the story very much more complex and very much harder to predict. So here is yet another photograph of mussels. This one looks a little bit odd because it's actually very pinkish. And if we zoom in very close on this mussel, we see that the shell is full of these tiny little holes which are pitting it. And if we look under a very high power microscope, an electron microscope, we can see that these holes are driven by cyanobacteria. Cyanobacteria are just a type of bacterium which is able to photosynthesize. In other words, it can trap sunlight energy and turn it into food. But in order to do that, they need something out of the shells. They need a source of calcium carbonate, and mussel shells are made of calcium carbonate. So in drilling into the shell of mussels, the cyanobacteria weaken the shell, and in the course of time, this can cause death to the mussel. So here's a nice clean shell, which is not attacked by cyanobacteria. Here we see the beginnings of attack. It gets worse and worse, and eventually the shell collapses and the mussel dies. And the rates of infestation by cyanobacteria can be very, very high. On this coastline, it can be anything up to 80% in a population. But in burrowing into these shells, the cyanobacteria do something else. They redeposit calcium on the outside of the shell, and that changes the color of the shell. It changes what's called the albedo, which is the reflectivity of the shell. That's the efficiency with which the shell reflects sunlight energy. So if we then take a thermograph of the temperature of these animals, we see that the animal which is covered by cyanobacteria is very much cooler than the animal which is not covered by cyanobacteria. So there's a real change in body temperature depending on whether you have cyanobacteria or not. And this can be very important. Here are the results for survival on this axis of the brown indigenous mussel and the blue invasive Mediterranean mussel during a heat wave that lasted for about 2 weeks. Here we have survival. Here we have the 50% survival cutoff. And we can see that mussels without cyanobacteria had well below 50% survival, whereas for animals with cyanobacteria, infested with cyanobacteria, the survival rate was closer to 70. And the effect was even more marked in the case of Mytilus galloprovincialis, the invasive species, possibly because it's a much darker color. So if you're looking at the relationship between mussels and cyanobacteria, if you're thinking about mortality because of shell collapse, having cyanobacteria or being infested by cyanobacteria is a really bad idea. But if you're worried about death through heat stress, then actually having cyanobacteria can be an advantage. And of course, the balance between heat stress and shell collapse is going to change under a regime of environmental change or global warming. So the story is not necessarily as simple as we might like to think it is. So understanding marine biology, I think, is essential to understanding why our environment looks the way it does, managing it to keep it looking the way it does, and basically critical to looking after our living resources. But personally, I'm much more interested in the fact that understanding marine biology helps us to understand how the natural world works. I want now to think about ecology a little bit more generally. And the first thing I'm going to say is that ecology is interesting because ecologists are often accused of having physics envy. And we're accused of having physics envy because there are no laws in ecology. In physics, we have Boyle's law, we have Charles' law, we have the laws of thermodynamics. We have all sorts of laws. But in ecology, there are no laws. Everything is conditional. Everything is context dependent. And there are a number of reasons for that. I actually think that rather than being like physics or chemistry, I think ecology is more like economics or more like sociology. Ecosystems are subject to multiple factors, and these factors interact with one another. We see threshold effects, which I'll explain in a moment, and we are constrained by our sample sizes. So when a chemist mixes two substances, they are dealing with a multiple of Avogadro's number. And Avogadro's number is the number of atoms in a mole of a substance. And it's a huge number. It's 6.02 times 10 to the power of 23. And if you're not sure what that means, it's 6.02 times 1 followed by a whole lot of zeros. So when you look at how two chemicals interact, you've got a sample size of some multiple of Avogadro's number of molecules which are interacting. You're looking at real average effects. What is the average behavior of all these atoms? In ecology, we have sample sizes of tens, perhaps hundreds, and occasionally thousands, but nothing like Avogadro's number. So in the case of ecology, we tend to deal not with true mean effects, but actually really we deal a lot with variability and with variances. So sample size is a big issue for ecology. We also have what are called threshold effects, and these can involve teleconnections. Teleconnections are where something very distant and unexpected influences the system that you're looking at. And this is an interesting example. It's an economic example. This is the exchange rate between the South African rand and the British pound. And you can see there's a threshold here where it suddenly dropped off on the 8th of May in 2006. On the 8th of May 2006, the then president of the country, Jacob Zuma, was acquitted on a charge of rape at 12 p.m. Now, this had nothing to do with macroeconomic policy in the country. It had nothing to do with the financial stability of the country, and yet it resulted in a dramatic change in the exchange rate. And this sort of threshold effect and these sorts of unexpected teleconnections actually occur quite commonly in ecology, and they're extremely hard to predict. Here's a nice example from the fishery in Egypt, where they were catching sardinella in the Eastern Mediterranean. And you can see the catches are very consistent for years, around 15,000 tons a year, until 1964, when suddenly it dropped to about a third of that, 5,000 tons a year. And then it dropped even lower in 1966. And this coincided with the opening of the Aswan Dam. The Aswan Dam is a huge dam which blockades the the River Nile and influences how much water can get down into the Mediterranean. And you can see the Aswan Dam was closed off in 1964 and resulted in a huge drop-off in the amount of fresh water which was being discharged by the Nile River into the Mediterranean. It changed the discharge of fresh water by so much that the salinity in the Eastern Mediterranean changed and all the fish moved away. So there's a very good example of a threshold effect and of a teleconnection. You build a dam in the desert several thousand kilometers from the coast, and the fish in the ocean swim away somewhere else, and your fishery collapses. These things are really easy to interpret in hindsight. They are extremely difficult to predict. You usually do not see them coming. In ecology as well, we have a huge emphasis on scale dependence. I want to elaborate a little bit about that. When you make an observation in ecology, the pattern which you see depends on the scale at which you make your observation. And in case that sounds a bit complicated, let me give you a very simple cartoon to illustrate. Each of these black dots is meant to represent the position of an animal. So here are 5 animals, and they're sitting together, and you can see that their distribution is basically random. They're just randomly dotted across the screen there. But if I zoom out and change the scale at which I make my observation, you realize that actually they're scattered, they're randomly distributed, but within clumps. There's one clump over here, there's a completely different clump over there. And if I change my scale of observation still further, what I see is that what I've really got are randomly distributed animals within clumps, and the clumps themselves are regularly distributed. So my perception of the pattern which I'm seeing in nature depends on the scale at which I'm making my observation. But it's not just the pattern which depends on the scale of observation. The processes which drive the pattern, the ecological processes driving the pattern, are also different. Those of you who are from Grahamstown and from Rhodes University know that Rhodes has got a really proud tradition in the field of rowing. And here is a picture of a chap, his name is James Thompson, and he was a Grahamstown rower, and he, with these other 3 guys, won a gold medal at the Olympics. That's quite an achievement. So someone from Grahamstown got a gold medal at the Olympics in rowing. And what you can see with all these chaps is that they're all jolly fit and tough and strong, and they're really ripped. Why are they so strong? They're so strong because moving a boat through water at high speed is very difficult. Why is moving a boat through water at high speed difficult? And the answer is that when an object moves through water, the molecules of water closest to the object stick to it. And the next molecule away sticks to them, and the next molecule away sticks to them. And that creates a force which slows you down. It's called drag. So objects moving through the water have a layer of water which is stuck to them, and that forms what's called a boundary layer. Now here's another cartoon, I beg your pardon. Another cartoon, this is of a phytoplankton cell. It's basically a single-celled plant which photosynthesizes. It performs the same function in the sea as grass does on land. It underpins the entire ecosystem. Here's the cell in green. Here's its boundary layer in pale blue, and it's sucked up all the nutrients out of that boundary layer. Now, the rate at which this phytoplankton can grow depends on 2 things: how much light it gets and how much nutrients it can get. And the light it gets depends on whether it's deep in the water column where it's very dark, or up near the surface where it's light. The amount of nutrients it can get depends on how quickly nutrients can diffuse across this boundary layer. So at very small scales, this is sub-millimeter, way sub-millimeter scales, the rate at which this organism can grow depends on the rate of diffusion of nutrients across the boundary layer diffusion rates. At a different scale, the rate at which it can grow depends on how much water is in the water column. It can be up near the surface where there's lots of light, but most of the cells are up there and the nutrient levels are low, or deeper down where there's less light, but lots of nutrients. So the ability of the cell to move up and down through the water column by changing its buoyancy will influence how fast it can grow. And at yet another scale, how much nutrient is in the water column depends on very large scale processes of kilometers, which bring very deep cold water from the depth of the ocean towards the surface where it can be used by phytoplankton cells. And that's the process of upwelling, which causes the west coast of this country to be so nutrient rich and the water so cold in the Benguela system. So very different processes control the patterns which you see descending depending on what scales we're working at. And in the sorts of work that we do, we work across multiple scales. We work at, I guess it's an abstract scale where one can do modeling. For example, you sit in front of a computer and you model how energy will flow through a system. How does energy get from those phytoplankton cells in the water column to those penguins which are breeding on Marion Island? And that would be one scale at which we can work. We also work in other small scales in lab studies. For example, here's an ex-student working in a laboratory in Hong Kong where he's studying proteomics, and he was looking at the expression of proteins by animals which have been subject to different stresses. So that's pretty small scale stuff as well. We also do field work where we do field manipulations, where we do things like we use cages and fences to exclude predators, to exclude grazers, and then we look at how that changes the ecosystem. So that's a rather larger scale, a whole organism scale. So we've gone from the abstract modeling to the molecular scale to the whole organism scale. And then we also work on very large scales where we look at systems across multiple countries. So we do work where we go to Morocco, Oman, South Africa, Brazil, and we compare ecosystems across these different continents and these different oceans. So we work across many, many different scales in order to understand marine biological ecosystems. But whatever scale we work at, we have a huge emphasis on experimental design and hypothesis testing. And this is an arbitrarily chosen picture of the design of an experiment we ran. We were looking at competition between the invasive mussel Mytilus and the local mussel Perna, where they compete with each other, and where Perna competes with itself, and where Mytilus competes with itself. The details are completely unimportant. What's important is that when you set up an experimental design, you need to pay very careful attention to the treatments that you have. Do you have the correct treatments? Are they unconfounded? Do you have controls in place? Do you have procedural controls in place? Do you have interspersion of your treatments? Do you have decent replication of your treatments? And most importantly, do you know how you're going to analyze the results? So before you set up an experiment, you really need to have thought out, how am I going to collect the data in such a way that I can analyze them using this approach, or will I have to use a different approach? And these are all things which you need to think about really carefully before setting up an experiment. I'm going to close with one last slide, and it's a very abstract one. It's actually a model of carbon monoxide concentrations in the atmosphere. And you can see the Southern Hemisphere does pretty well. Africa does pretty well. The worst concentrations are over the industrialized nations, China, India, Europe, and United States. But I'm not going to talk about carbon monoxide. I want you to think of this more abstractly, in my case, as the balance of nature. Why does nature look the way it does? So this is a very abstract concept. Hopefully you can apply it to whatever discipline it is that you work in. Why does the system I'm studying look the way it does? And every time we run an experiment or we do a study, we open a window onto this, which helps us to understand why the system we're studying looks the way it does. It might be an ecological system, a sociological system, an economic one, whatever it happens to be. Importantly, in ecology, the window which you open gives you an insight which depends on where you open your window. If you open it somewhere else, you may get a different understanding of how the ecosystem works. And of course, the balance of nature is not static. The balance of nature is highly dynamic. So your understanding of the system depends not only on where you do your experiments, where you open this window, but when you open it. Is it now or is it now? And that will influence how you understand the system that you're studying. But I think most important of all for me, the real take-home message from this figure is that you do need to recognize that the window which you open up onto the system you're studying is all that you have to work with. So it's not a good idea to be too dogmatic about my understanding of the system is the only correct one. Thank you.
Wow.
Sue, I'm done. What happens next?
Yeah, thank you so much. I found that absolutely fascinating. And I'm sure that others did too, regardless of whether they come to us as biologists or as someone like me who's much more on the sort of sociological side of things. It actually fascinated me how much of what you said was very clearly linked to sociological issues. You were looking at your phenomenon with through biological lenses, hugely important, but it's really important that we can speak to each other. And Kevin, I see, is saying he's a non-biologist and he feels well enlightened. Thank you. What I'm going to ask people to do now is either put your questions in the chat if you would like to type your question, or if you would like to ask a question, please just put the letter M for microphone. And if you put the letter M into the chat bar, then I will call your name and you can turn on your microphone and ask a question. So just to repeat, please can you type a question into that chat bar and Christopher can tackle it, or put the letter M if you would like to ask a question. In the meantime, I'm going to have a look at some questions that have come up so far, or that I jotted down myself, or people sent to me privately in the chat. And while I do that, please just go ahead, the rest of you, and type in your questions or comments. So one of the things that I thought was really interesting, well, there was a couple of things that I thought was really interesting, but one is that whole idea that you only see what you're looking at. And I think that was so clear to me for whatever kind of research you do. You need to have a sense of what it is you're looking at. And I think this last Balance of Nature showed that, but really all the way through, to really have a sense as researchers of the scale at which you're looking at something, because whatever scale you're looking at, it is really going to affect what you can see. And so we have to be very careful as researchers of assuming that our conclusions offer a full account of the phenomenon, or that our conclusion on one scale hold for another scale. So you gave us different scales in your field, which was modeling, molecular work, sort of field work, organism scale, and then large scale system-wide. What is your favorite kind of work?
My favorite is the manipulative field experiments. And that's because it's because you have control over what happens. Molecular work, yeah, you end up sitting in a laboratory all the time, and that doesn't inspire me. Looking at large-scale systems is great, and it's really fun, but and it's probably the most pleasant to do, but if you really want to understand something, a manipulative field experiment is the best way to do it. So you can actually control all the different variables and get a much better understanding of the system than if you just make observations. And that, I think, is one of the advantages that we have in biology, that we're able to do that to a degree which in other disciplines it's not possible for ethical or practical reasons.
Right. Yeah. On that ethical issue, should we be not eating mussels now? Do I have to give up mussels?
No, not at all. As long as you're not part of a huge crowd of people who are massively overexploiting, it's simply not a problem. The sorts of numbers of mussels which you're legally allowed to collect yeah, that's perfectly sustainable.
Great. And for me as a sociologist, I know that you were saying it's not your particular interest, but certainly you articulated very clearly, is this whole relationship between that any kind of true sustainability is probably not the small scale fixing, although I found that very interesting, the way you put that grid over to allow the mussels to grow. But the real solution to our plundering of the planet would have to be a socio-economic one that took care of people's basic needs so that they didn't have to go and plunder. Someone sent me a private comment just saying, you know, that the problem is, of course, it's poor people, you know, sort of being not blamed, because you certainly weren't blaming, but sort of that are in this case clearly the problem in terms of mussel, and needs must, you know, if you're hungry, you need to eat. And the same person just asked me, is there mussel farming? Is there sort of big industry mussel farming, or is there no such thing?
It Yeah, there is, but it depends on the country. There isn't in this country because it's too wave exposed, and there are very few places where it's possible. There's some mussel farming in Saldanha Bay, which is a very sheltered bay. Otherwise, it's not possible on this coastline. Other places like Scandinavia, Spain is a very big mussel farming industry. It's the Chilean coast. Japan, a lot of countries do farm mussels, but the problem is absolutely not an issue of blame at all. On the contrary, it's one has to feel sorry for these people that they have no recourse except to do this. And ultimately the problem is that we have too many people on the planet with too high a level of material well-being, essentially.
Can you just say a little bit more about that? So it's about overuse or overconsumption of planet's resources by a few. Is that what you're saying?
No, it's actually overconsumption of the planet's resources by a lot of people. There are, I don't know, I have no idea what the numbers are, but there are far more people than there used to be, let's say even 100 years ago. And each of those people now places much more pressure on natural ecosystems than people would have done 100 years ago. So it's a combination of our standards of living, if you like, and our levels of consumption, directly or indirectly, and just the sheer number of people.
Right.
Yeah, so, and that's, but there's also, I have a slightly different take on these sorts of things, perhaps from other people, which is, if mankind as a species were to go extinct, well, that from the planet's perspective, that would be a really good thing. And also, sometimes we say, oh, well, the planet will change to this, that, and the other. And we say, well, there's nothing here but cockroaches. And the answer is, well, yeah, we don't like cockroaches, but the planet doesn't care whether it's people or cockroaches. So yeah, we have a very anthropocentric, unavoidably anthropocentric perspective on what's good for the planet.
Yeah, absolutely. A few questions here.
I feel like
one of the questions is, what is the name of the strange creature that can eat, the bizarre creature that can eat bigger than its head?
There are a number of different fish that are deep sea fish that can eat things bigger than they are. It's really bizarre, but it's true. They have very extendable stomachs. They have enormously openable jaws, and they can swallow things bigger than they are. It's absurd.
That is fascinating.
But again, needs must.
Yeah. What is a caracule? Someone is saying, I live in Quilera Mouth and there's a lot of poaching of mussels, but I'm not always sure which species of mussels are being poached. And then the question is, what is a caracule?
I have no idea what a caracule is.
Alecrucal.
Which river mouth did you say?
Quilera.
Oh, alecrucal. Alecrucal is a type of snail, basically.
Okay.
That's near East London. Yeah, that will be the brown mussel perna perna, the native mussel. The Mediterranean mussel doesn't go very much past Port Elizabeth. It does get up to just into Transkei, but only in very low numbers. And alecrucal is basically a rather large marine snail.
Right. And then the next question is about, is the waste products from mussels being tested for capacity for production of energy, i.e. in digesters?
Sorry, could you say that again, please?
It's a question about whether there is a benefit to using, let me read it exactly. Has the waste from mussels been tested for a capacity to produce energy, i.e. in digesters?
No, it hasn't. I mean, it's actually an interesting question because mussels produce feces like other animals, but they also produce something called pseudofeces, which is they are filter feeders, so they're extracting material out of the water column, but they then sort it. And the particles which they think they can digest, they will consume, and what's not digested will form feces. But what they think they can't digest, they will set aside and produce what are called pseudofeces. So they haven't actually, they're waste material that hasn't actually gone through the gut. But as far as I'm aware, no, nobody's ever had a look at that.
I must say, I find it
I
interesting
feel like
things are being used nowadays for.
I feel like this yes. This is a bit like being on a ship in the olden days when you used to have to say over, meaning it's your turn.
Indeed, it is rather strange. And then someone else saying, and this is an interesting point that Anzio is making from Two Oceans Aquarium. And Anzio is saying, interestingly, the increase in the invasive mussel species correlates with an increase of endangered African black oyster catchers, which feed on them. So there's always a silver lining.
Yeah, that's correct. And the reason is that the invasive mussel is able to colonize more of the shore than the native species could. So there are now more mussels, and that has translated into more oyster catchers. So yeah, absolutely a silver lining.
Yeah, these things are never straightforward and just, yeah. So Robert says, a great shout out to all those doing so much to preserve our biodiversity. A great talk. Thank you, Professor Christopher. And I think on this note, unless anyone else has any last questions, I think we could probably wrap it up and just say thank you so much. I found that absolutely fascinating. It was a little bit like watching a nature program, but much better because it's real people doing real work. Not that the people on the TV aren't, but people that we can actually know and. And someone actually said, I'm trying to see now who it was, someone actually said, hey, that's me, when one of the pictures came up. It was
really?
Francesca Pori, who said, hey,
Francesca.
that's me. Yes,
Francesca.
So,
ciao, Francesca.
Wow. So it's really lovely to have had you with us this afternoon and to hear about your amazing work. And yeah, thank you everybody for joining us from across the planet. It's a very diverse group of people from Kenyan, Ghanaians, Swedes, all sorts of folks. But I think with a common interest in, as you started the talk by talking about the sort of love of science and curiosity-driven science. And I'm going to end off doing my own little political soapbox thing again. Yeah. There's so much pressure on universities to think about our science and our research in terms of immediate commodification of knowledge. And I'm not saying we shouldn't be doing that. I'm not saying we shouldn't be doing applied research. But I loved your point that, you know, a lot of industry is doing research and development in those kinds of applied ways. And it really is, the universities are the last spaces that can be driven by purely curiosity-driven research, and we need to hold on to that. Your research is clearly applied. It's clearly going to be hugely beneficial to the planet, but it's also just beneficial to us to drive our curiosity. And I think that was clear from this afternoon by how diverse the group was and how much everyone enjoyed it. I'm going to leave over with hand over to Simone for one last question. Go for it, Simone. Grab the microphone. You can turn your mic on and ask us the last question. If you can find her microphone button, Simone.
Hello,
yes.
Can you hear me?
Yes.
Oh, Christopher, it's me, Simone from Chile. How are you?
Hey, Baldanzi! Ciao!
Ciao, Christopher. It was
yeah,
very nice
good to hear you.
Yeah, sure. Good to hear you too.
No, thank you.
It was a very nice talk, and I wonder if currently or recently, have you ever touched any of the I would say any investigation and any research on pollutants and mussels, and I'm referring particularly to microplastics.
Yeah, we have. We've been doing a little bit of work on microplastics, but for me, even more interesting, we've been looking at the effect of plastic leachates on spatial patterning in mussels, and that's been really, really interesting. We're working on a couple of manuscripts with that at the moment. Yeah, are you working on that?
I'm planning to.
Good.
And we're still applying to some funding for this research. So I was interested on it. It's quite unknown field. There is lots of work on mussels, especially in Europe and in China as well. All of this very place in the earth where there are lots of pollution, but that's still a lack of how microplastic can be distributed and the occurrence and the relationship with big oceanographic features like upwelling, for example. So that's still lots to do in this field, and it's quite interesting. So yeah, thanks again.
I think not sure. There's a lot of potential there. Good luck with the funding.
Great. And I see Anzio has said that someone at Two Oceans Aquarium has in fact done a study on microplastics in mussels. So it might be nice to connect there. Oliver asked the question, have you, Christopher, or any of your team looked at heavy metals in mussels?
Not in mussels. We've looked at heavy metals and other things, but not mussels particularly.
I have to say at this stage, I don't really even know what heavy metals are, but I'm sure they're really bad things. I'm presuming they.
Don't worry about it.
Metal I know is head banging stuff, but heavy metals are very bad. Florence tells me bad.
Yes. That's what he means.
Thank you so much for a really enjoyable afternoon. Thank you everybody else for joining us. If you want to hear about what's coming up, join.
Thanks very much.
Thank you, Christopher. It really was great.